Good day everyone, I am Rosen Romero from Set 3A. So for today's video, we're going to tackle the Unit 12, the gene expression. Here's our topic outline.
12.1 Control of gene expression in bacteria. 12.2 Structure of the nuclear envelope. 12.3 Packaging the eukaryotic genome. 12.4 Heterochromatin. 12.5 The structure of a. 12.6 The human perspective, chromosomal aberrations, and human disorders. 12.7 Epigenetics. There's more to inheritance than DNA. 12.8 The nucleus as an organized organelle. 12.9 An overview of gene regulation in eukaryotes. 12.10 Profiling gene activity. 12.1 Basic concepts.
Expression in bacteria is tightly regulated to ensure efficient responses to changing environment. This control involves both positive and negative regulation mechanism. Transcription factor play a key role in this process. The first one is the operons. Bacterial genes are often organized into operons, where multiple genes are transcribed together. The lack operons, for example, regulates the metabolism of lactose in E. coli. The second one is the repressor proteins. Negative regulation occurs when a receptor proteins bind to the operator regions of DNA, preventing RNA polymers from transcribing the genes. The lack receptor inhibit transcription in the absence of lactose. The third one is inducers and corpressor. Small molecules like inducers, example lactose, or corpressors can affect the activity of repressor proteins influencing gene expression. The fourth one is activator protein. Positive regulation involves activator proteins that enhance binding to a specific DNA sequence. They can be influenced by small molecule as well. The fifth one is the sigma factor. Different sigma factor guide RNA polymer to specific parameters, allowing bacteria to respond to various environment conditions. And the last one is riboswitches. These are regulatory elements in the mRNA molecule that can be directly bind small molecule influencing transcription or translation. Bacterial gene expression is regulated through operons, receptor proteins, and activator proteins. Operons organize genes, and receptor inhibit transcription by binding to operators. Inducers and corpressors affect receptor activity. Activator enhance transcription, and sigma factor guide RNA polymers and promoters. Ribosome switch in mRNA directly respond to small molecules, influencing gene expression for bacterial adaptation.
12.2 Structure of the nuclear envelope.
Nuclear envelope consists of two membranes, the outer nuclear membrane and the inner nuclear membrane, which enclose the nucleus in the eukaryotic cell. Here are the key pictures including double membrane. The nuclear envelope is double membrane structure, which an outer membrane facing to the cytoplasm, while an inner membrane facing the nucleoplasm. Second one is nuclear pores. These are proteins-lined channels that span both membranes, facilitating the exchange of materials such as RNA and protein between the nucleus and the cytoplasm. This is a mesh-like network of protein filaments lining in the inner surface of the inner nuclear membrane. It provides structural support to the nucleus. The fourth one is prenuclear space. This is a space between the inner and the outer nuclear membrane, serving as a buzzer zone. This nuclear envelope acts as a barrier separating the genetic material within the nucleus from the cellular cytoplasm. It plays a crucial role in regulating the passage of the molecule in and out of the nucleus, contributing the cellular function and maintaining the genomic integrity.
12.3 Packaging the eukaryotic genome.
The eukaryotic genome is efficiently packaged within a cell nucleus through a hierarchical structure. DNA wrapping, DNA, is initially wound around the histone protein to form nucleosomes. Nucleosomes consist of DNA colloid around the octamer of histones, creating a bread-like structure. Chromatin fiber. Nucleosomes further coil and fold to form a more condensed structures known as chromatin fiber. This compaction helps to manage the genome size within the limit space of the nucleus. Chromosomes. As cells prepared for cell division, chromatin undergoes additional level of folding and coiling, ultimately forming the strict structure called chromosomes. Each chromosome contains a single long molecule with a specific genes. Centromeres and telomeres. Centromeres are the region on chromosome crucial for proper segregation during cell division. Telomeres are the protective caps at the ends of chromosomes, preventing degradation and ensuring genomic stability. This packaging is not only fits the lengthy DNA into the nucleus but also play a role of gene regulation. Chromatin structure influence the accessibility of gene to transcriptional machinery. In fact, gene expression, the dynamic nature of chromatin allows cells to regulate gene activity in response to environment cues and developmental needs.
12.4 Heterochromatin.
Heterochromatin is a condensed and tightly packed form of chromatin found in the cell nucleus. It has a distinct structure and associated with the reduced genes expression. Key characteristics of heterochromatin including condensed structure. Heterochromatin appears as dense, tightly coiled region of chromatin concentrating with a more dispersed and open structure of euchromatin. Gene repression. Genes with the heterochromatin are often transcriptionally inactive or expressed at low levels. This repression is due to the compact nature of heterochromatin, limiting the accessibility of the transcriptional machinery to the DNA. And the third one will be the late replication. Heterochromatin is typically replicated later in the cell cycle compared to the euchromatin. Position in nucleus. Heterochromatin is often located near at nuclear periphery, and it can form distinct region known as heterochromatin domains. Epigenetic modification. Chemical modification to histone protein and DNA, such as methylation, contribute to the establishment and maintenance of heterochromatin. Heterochromatin is densely packed and traditionally inactive of chromatin, found in the cell nucleus. It is characterized by its condensed structure associated with the gene repression, late replication in the cell cycle, peripheral nuclear location, and epigenetic modification. Heterochromatin plays an essential role in maintaining genome stability and regulating gene expression.
12.5 The structure of a mitotic chromosome.
Chromosomes undergo structural changes during cell division. Chromatids. Each mitotic chromosomes consists of two sister chromatids connected by the centromere. The chromatides are genetically identical and result from DNA replication during the S phase of the cell cycle. The second one is the centromer. The centromer is the specialized region that holds sister chromatides together. It plays a crucial role in ensuring proper segregation of chromosomes during the cell division. The third one will be the kinetochore. The kinetochore is a protein structure associated with the centromer. Serve as the site of microtubule attachment. Microtubules, parts of the cell's cytoskeleton, interact with the kinetochore to facilitate chromosomes movement. Condensation. Prior to mitosis, chromatin undergoes condensation, transforming into visible and distinguishable structure. This condensation is essential for efficient chromosome segregation. The last one is the X-shaped appearance. As chromosomes condense, they often exhibit an X-shaped appearance reflecting the paired sister chromatids held together at the centromere. The structural organization of mitotic chromosomes ensures the accurate distribution of genetic material to the daughter cell during the cell division. This process is crucial for maintaining genomic stability and cellular function.
12.6 The human perspective, chromosomal aberration, and human disorder.
Chromosomal aberration structure or numerical changes into chromosomes are likened to the human disorder. Example including Down syndrome, trisomy 21, Thunder syndrome, monosomy X, and Klinefelter syndrome XXY. Specific chromosomal deletion as seen in Cry-Duchamp syndrome also lead to the distinct disorder. Chromosomal aberration are implicated in the various cancer, emphasizing their significance in both genetic research and clinical application for diagnosis and the genetic counseling.
12.7 Epigenetics. There's more to inheritance than DNA.
Epigenetic explores heritable changes and activity without altering DNA sequence. DNA methylation and histone modification influence gene expression, while non-coding RNA molecules regulate translation. Epigenetic changes can be inherited across generation and are influenced by environmental factors, coordinating our understanding inheritance beyond DNA and emphasizing the dynamic interaction between genes and the environment.
12.8 The nucleus as unorganized organelle.
The nucleus, a vital organelle in eukaryotic cell, is organized with distinct structure and function. Nuclear envelope. Comprising inner and outer membrane, the nuclear envelope encloses the nucleus, separating genetic material from cytoplasm. Nuclear pores facilitate communication between nucleus and cytoplasm. The second one is the chromatin. DNA is organized into a chromatin complex of DNA and protein, or we can say histones. This dynamic structure can be accessed in euchromatin, open and active, or heterochromatin, condensed and inactive, regulating gene expression. Nucleolus. Within the nucleolus is the specialized region responsible for ribosome synthesis. It contains rRNA, or we can say ribosomal RNA, and the protein contributing to cellular protein of production.
12.9 An overview of gene regulation in eukaryotes.
Epigenetic explores heritable changes in gene activity without altering DNA sequence. DNA methylation and histone modification influence gene expression, while non-coding RNA molecules regulate translation. Epigenetic changes can be heritable across generations and influenced by the environmental factors, bounding our understanding and inheritance beyond DNA and emphasizing the dynamics interaction between genes and the environment.
12.10 Profiling gene activity.
Gene activity involves assessing gene expression levels on a large scale. Techniques like microarray, RNA sequence, or RNA-seq, quantitative PCR or qPCR, and single-cell RNA sequencing offer diverse approaches. This method provides insight into gene regulation, biological processes, and diseases mechanism, aiding in the identification of potential therapeutic targets.